Hey guys, I'm going to use this slideshow as an opportunity to try and condense and summarize a huge part of this course, which we did a whole chapter on called cellular respiration. So, what I want you guys to think about as we go through each of these slides is that cellular respiration is a huge umbrella term describing a lot of chemistry happening inside of living cells to get energy from food to little tiny molecules called ATP. And ATP, remember, is the molecule that cells use to make endergonic reactions happen. So cells need a constant supply of ATP to do everything, to do all the work that they need to do. Now, keep in mind that cellular respiration is an example of a very complex metabolic pathway that involves lots of different enzymes and lots of different substrates. And these substrates are being changed into products that then become substrates for other enzymes. So it's a lot like a very complicated highway system with lots of entrance ramps and exit ramps with things coming on and things leaving, and it's all interconnected. It's really difficult to understand the significance of these processes by just looking at one car on the highway or one particular pathway. It's all interconnected. But the main idea is to get energy out of food and get some of that energy to these tiny little molecules called ADP and change them into ATP. And that energy can then be used to do cellular work. Now, cellular respiration overall is a catabolic process, which means, if you guys remember, catabolic means a process that's breaking big molecules down into smaller molecules. And it's an exergonic process, which generally means it's releasing heat in some way. And it's releasing energy, almost always in the form of heat, but maybe possibly transforming some of that energy into ATP energy, which can be used to do cellular work. Now, cells can make ATP a couple of different ways. And I'm going to break these up into direct versus indirect. Now, the direct production of ATP happens when an enzyme transfers energy from a food molecule directly to ADP and in the process phosphorylates it to change it into ATP. It's using enzymes. And indirect ATP production involves a lot of complex structure. And we're going to talk about that when we talk about chemiosmosis. So I'm going to save that for later. Now let's focus first on direct ATP production, often referred to as substrate level phosphorylation, okay? Because there's that word substrate, okay? That tells you it involves an enzyme. So substrate level phosphorylation means you're changing ADP, okay? You're changing ADP into ATP by using energy from food. And you're doing this directly with one enzyme. It's a simpler process, okay? Let me get rid of all that. All right, now it's direct because this enzyme changes its shape. It's able to do this because of the way it's built. It's folding, its three-dimensional structure, as it interacts with a food molecule, a substrate, and in the process, phosphorylating ADP. The hopefully most familiar example of substrate level phosphorylation is glycolysis. And if you guys remember, glycolysis takes place in the cytoplasm, and it's the process, the metabolic pathway that converts glucose into pyruvate. It only produces a few ATPs, so it's not as efficient as indirect, but we'll talk about that later. It's also, most importantly, anaerobic, which means it doesn't require any oxygen. So direct ATP production, like glycolysis, was happening before oxygen was a player on the stage, so to speak. Now, the Krebs cycle, aka the citric acid cycle, also does some direct ATP production. This happens inside the mitochondria, and in this case, pyruvate is being broken down to produce carbon dioxide. And this also produces just a few ATPs. And finally, there are these fermentation processes. And fermentation, if you guys remember, is another cytoplasm process that converts pyruvate into other stuff, okay? Not carbon dioxide directly, but changes pyruvate into other metabolites or products, and in the process allows the cell to make a few ATPs. So direct ATP production is pretty straightforward, almost always happens in the cytoplasm or inside of mitochondria, which remember used to be free-living aerobic bacteria, so that would be their cytoplasm if you go back in history. So let's now look at indirect ATP production, which I think is one of the hardest things we tackled this year. And I'm going to do this two ways. I'm going to do it using words on this slide, and then we're going to do it pictorially like we did on the big sheets, if you remember in class when we drew those big sheets. Okay, so it's called indirect ATP production because the energy isn't flowing directly from the food molecules to ADP to make ATP. There's some intermediary steps that involve structures like membranes and compartments and gradients. And in this case, we're setting up a gradient between two sides of a membrane, and this gradient is able to store potential energy. Now, we called this gradient energy system the electron transport chain. And if you remember, it's embedded in the membrane that separates the inner and outer parts of mitochondria. And these compartments are absolutely required. If they're punctured or torn or ripped, the gradient disappears because diffusion just equalizes everything and it won't happen. They also require oxygen, and oxygen is going to be accepting electrons from the chain and becoming reduced and changing into water. It requires a steady supply of electrons that are delivered to it by carriers, NAD and FAD, which I'm hoping you remember. They have kind of silly names. And what's happening is we are passing electrons from carrier molecule to carrier molecule, and as they give up their energy, that energy is being used to force hydrogen ions from one side of the membrane to the other. Now, if you remember, the hydrogen ions are being pumped out of the matrix into the intermembrane space. That's even more points. Okay. Now, the payoff comes when the hydrogen ions flow backward back into the matrix through an embedded protein or enzyme called ATP synthase. And this backflow is where most of the ATP production is coming from. So let's try to relate this now to a picture. So if you guys look at this picture, we have a big oval cell here with lots of cytoplasm. So all this stuff here is the cytoplasm. And in the middle, okay, I have drawn an oval which represents our mitochondrion. Now the mitochondrion has an inner part called the matrix, which I've colored blue, and an outer part, which I've called the IMS, or the intermembrane space. And it's funny, on my phone, which I'm using to control this presentation, it looks orange, but on my laptop it looks green. So I don't know if that's just me or my computers. But if you guys remember, what's happening here is glucose is being broken down into pyruvate, okay? And pyruvate is being broken down into carbon dioxide. And in these two processes, we're capturing high energy electrons and giving them to carriers called NADH and FADH2, these electron carriers. And these electron carriers, what they're doing is they are delivering electrons to the beginning of the electron transport chain, which I've represented here with these little pink dots. And as the electrons are flowing from carrier molecule to carrier molecule, they are causing the pumping of hydrogen ions from the matrix outward into the intermembrane space. Okay? Oh, I have ATP synthase in the wrong place. ATP synthase should be embedded in the membrane between the matrix and IMS. So I'm going to draw it again here. So here's ATP synthase. Boom, boom. And so the hydrogen ions are then flowing back into the matrix. And in the process of flowing back into the matrix, they allow ATP synthase, just as its name says, to convert ADP into ATP and make lots and lots of energy. Completely ignore that. That is wrong. ATP synthase is not there. It's located on the boundary between the matrix and the intermembrane space. There you go. Classic example of a teacher teaching himself as he goes through this. So I hope that helps you make sense between this picture and the slide before. If it doesn't, Go back to your big sheet or go back to your textbook, take a look at it. But remember, the little details are not important. It's where is the ATP coming from? And in this case, ATP is being made by a gradient set up between two different places inside of a mitochondrion. And that gradient is a hydrogen ion gradient. And as it tries to equalize using ATP synthase, it produces that ATP that every cell is after. That's the reason all of this happens. So let me clean up this, get rid of that. All right. So another pair of words that I've used a couple times already, want to make sure we focus on them, is aerobic versus anaerobic. Okay. Now, aerobic respiration requires oxygen. Okay, and it was named this before they even knew oxygen was the gas. Aerobic literally is an old word that just means air. But we now know that the important gas, the key to all this, is free atmospheric oxygen. And aerobic respiration is basically two things together. It's glycolysis plus the Krebs cycle or the citric acid cycle plus the electron transport chain. So all three of those things combined make up something we call, or that you may see referred to as aerobic respiration. It only works if oxygen is available, because remember, oxygen is needed by the electron transport chain to accept those electrons after they've given up their energy. It is the final step of the electron transport chain. And written chemically, it's half of an O2 molecule plus 2 hydrogen ions produces 1 water molecule. Now, anaerobic is the same thing as fermentation. These two words are basically interchangeable. And what that means is it's a way to take pyruvate and make more ATPs out of it without using oxygen. And remember, atmospheric oxygen is a relatively new player in the evolution of life. So there were lots and lots of living things that were doing cellular respiration a long time before oxygen was available. So that means their cellular respiration was limited to glycolysis and fermentation. Not true anymore. Now, because of this, all life on Earth has the enzymes and the DNA and the genes to produce these enzymes and run this process we call glycolysis. It's a very ancient process. Okay, now there are different types of fermentation pathways. We focused on a couple of them: alcohol fermentation and lactic acid fermentation. Okay, so when you see the word anaerobic respiration, that's the same thing as fermentation. Now, if we focus on ethanol fermentation, don't be intimidated by this. Let's just look at what you need to know from this. Ethanol fermentation basically takes a glucose molecule as the initial substrate, does some stuff to it, and down at the bottom produces ethanol. All right. So if you look all the way down here at the bottom, you see ethanol down here at the bottom. Okay. Now, as that's happening, if you look at this pathway right here, okay, as ethanol is being produced, NAD plus is being regenerated, and the NAD plus is needed to keep glycolysis going. So the extra ATPs are coming from the continuing functioning of the glycolysis pathway, because glycolysis needs to put electrons somewhere, and so they're given to NADH. And if all the NADH is filled up with electrons, glycolysis stops. So ethanol fermentation is a way for cells anaerobically to convert pyruvate into ethanol, and in the process keep glycolysis rolling along and making a few ATPs. Okay, lactic acid fermentation is a little simpler. You have the glycolysis part. Glucose is being converted into lactic acid or lactate, and in the process, just like we saw with alcohol fermentation, you're regenerating your NAD plus to keep the glycolysis part going. And if you notice, it's the glycolysis part that's making the ATPs. Okay, so that's it. I think that's a pretty good summary of what cellular respiration is, and I'm hoping you can apply this to questions about early Earth and the oxygen revolution that we talked about, and how cells, simple cells like bacteria, can get along just fine doing fermentation. The fancy way of doing things, chemiosmosis, you need a mitochondrion to do that, and that's something that animal and plant and fungi cells do all the time now, because oxygen currently makes up about 20% of our atmosphere. It's a much more efficient process. So this chemistry, this biochemistry touches on a lot of the aspects of this course that might be woven into some questions you're asked on the AP exam this year. Only time will tell, but I hope this helped you sort some of it out. Stop in there. Thanks, guys.